Okay, so we've got a picture here of transfer RNA, which you're probably familiar with. This four-leaf, this clover thing. So what we have here is we have these loops, and there's hydrogen bonding between complementary base pairs here, the RNA, there and there, and at the 3' end we have got the amino acid attached, and at this end, this is called the anticodon. So in this case, I've got 3 A's there, which would bond to the codon on the RNA, which on the mRNA, which would be UUU. So this amino acid, which actually be phenylalanine in that case. So we should be fairly familiar with that tRNA. And you may even have seen, I've taken this one. This is from the Wikipedia page. This is what the 3-dimensional structure it looks like. So you can see that those base pair regions form helices, and overall it's an L-shaped molecule. And here on the end there we've got, I think that's actually tryptophan, that amino acid they've drawn there. We've got the amino acid on that end there, and down here you would have the anticodon where it bonds to the mRNA. So how was this discovered then? Well, this was actually discovered in 1957, which is only a few years after the Watson and Crick model of DNA was proposed. And it was discovered by these 3 people, Hoagland, Stevenson, and Zemechnik in America. And if you want to, you can look up that paper. It's in the Journal of Biological Chemistry. You can get the whole thing free on the internet, a reprint of it. Right, so let's just think about where this idea came from. So, well, you know, Watson and Crick, obviously early 50s, they came up with the double helix model for DNA, and it was fairly obvious to a lot of people then that the base sequence in DNA had to be translated into amino acid sequence in protein. That's how the genetic material was stored. And also fairly quickly people came around to the idea of a triplet code where 3 bases represented 1 amino acid. And it was also realized then, in order to get from this base sequence to the amino acid sequence, you're going to need some kind of adapter molecule. And it was Francis Crick who was the first one to suggest that this adapter molecule was likely to be an RNA molecule, because he realized that probably some kind of complementary base pairing was the way in which the genetic code was preserved. You know, the order of the bases was then adapted into an order of amino acids. Base pairing would be essential, so it's going to be a nucleic acid, probably RNA rather than DNA. So that's where the thinking was in this sort of late 50s when this discovery of RNA was made. Now, how was it done? Right, now this group had been working on protein synthesis for a long time. Okay, so I'm just going to rub some of this out here. Right, they were looking at protein synthesis, and they wanted, they were the first, I think the first group who properly looked at it in vitro. So that means in a test tube, rather in vivo means actually a living cell. So they were able to break away from having to have living cells to show protein synthesis. They could recreate the chemical reactions in a test tube, this group. So how did they do it? Well, they got some rat hepatocytes, so that's rat liver cells, and they homogenized them, so they mushed it all up in a buffer. And so you've got like a whole mixture. And then this is a process here which you've probably come across before, called differential centrifugation. Sorry. Okay, so how does that work? Okay, So here, we've got the hepatocytes, we give it the first spin, quite low speed, and all the big bits come down to the bottom, and that is mostly going to be nuclei. They're not interested in that, so that is that pellet there is thrown away, discarded, and they keep this stuff, which is called the supernatant, so drawn in red, so the stuff that is still liquid, and this time they spin it again for a second time, but faster. Quite a lot faster, and then the next big, all the really big bits like the nuclei have gone, the next biggest bits are the mitochondria, and you get a pellet of mitochondria there, and they weren't interested in them, so they discarded them, threw them away, and they kept this, the liquid bit at the top, again it's called the supernatant, and then they gave it a third spin, this time a lot faster, okay, and you end up with a pellet, and here, which are called microsomes. They call it the microsomal fraction. And the microsomes, even though at that point people were not that sure about what ribosomes were, they knew there was some kind of RNA bit in the microsome. So this was, we now know it to be, it's ribosomes, mostly stuck to the endoplasmic reticulum, which of course membranes. So you've got a whole load of ribosomes there. And the supernatant here, well, that contained a whole load of other things, but it contained a lot of protein and some RNA in there. Right. So this group, what they did was they that before they were interested in transfer RNA, they were interested in protein synthesis. So I'll just show you what they did here. They got this whole lot, and they added radioactively labeled amino acids. So they got carbon 14, and leucine was the main amino acid they used. I don't know why they used that one in particular, but they repeated it with other ones, and they showed that if they added leucine to that, they needed to add some ATP. Not surprisingly, for providing energy, and a little bit more surprising, they found it didn't work unless they added GTP as well. And if they did that, they would end up getting a protein, and the protein contained some 14C from the radioactive amino acids that they'd added to the mixture. So they proved that protein synthesis was occurring there in vitro in the absence of living cells. And obviously, you don't need nuclei, you don't need mitochondria. You do need the microsomes in order for it to occur. They needed that, so they probably began to think that was the site of protein synthesis. But you also needed this supernatant to make it work, and they realized there were some proteins in there. Obviously, they're going to be enzymes probably that are needed, and there was also some RNA in there, and that was why they started focusing on the RNA. What was the RNA doing? Okay, so they've got an in vitro system for protein synthesis then. So I'll just rub that out. And now they started investigating it. So what they did was they got this supernatant here. So there's a supernatant which contains RNA and proteins, and they treated it. Right, in this, what they did here was they extracted all the proteins. So if you add phenol, you can get rid of all the proteins. So all you've got in that tube is the RNA, just RNA. They didn't get rid of just the RNA and leave the proteins, but they just left those two mixed up together there, the RNA and the proteins. Now, they called it RNA and proteins. What we would now know what they're talking about, we actually had transfer RNA. That's the kind of RNA you've got there. And obviously you've got transfer RNA here. And the proteins are actually enzymes. And the enzymes are responsible for joining on the amino acid to the transfer RNA. And those enzymes are now called amino acyl transferases. And there's quite a lot of them because each one is specific for the different RNA with different code on the different amino acid on it. So a whole load of enzymes called amino acyl transferases, they're responsible for joining the amino acids to the tRNA. They didn't call them that at that time, but that's what they are. Right, so let's see the experiments they did then to show about what was going on with the RNA. Right, now, first of all, we have to show that the RNA and the tRNA join together. So let's do that. So let's demonstrate, show that the tRNA, sorry, and the amino acid join together and are covalently joined together. So how did they show that? Well, they got the enzymes. So they've got the enzymes, these proteins here. They've got the enzymes, and they added the purified tRNA here, which they didn't know was tRNA, but that's what it was. And they also got some radioactive amino acids there. So that's labeled with 14C. amino acids. And they got a protein was made with the 14C incorporated in it. So they've got a radioactive protein made, so they know that protein synthesis has occurred. Now, in order for that to work, they had to add some ATP. So that experiment showed them that amino acids do form a covalent compound, if you like, with a small RNA molecule. This we now know it's transfer RNA. So they showed that. No one had shown that before. The amino acids physically binding to an RNA molecule, the adaptor molecule that everyone was interested in finding. They also said if you leave out the enzyme there, so if you leave that out, no protein synthesis. So, no radioactive 14, no protein with 14C in it. So that showed you that in order for the amino acid and the transfer RNA to join together, you do need an enzyme, and that enzyme was present in the supernatant there. And we now know that enzyme is an amino acyl transferase. So that was the first bit of evidence. The next thing that they had to do was they had to show, scroll back a little bit. They had to show that the tRNA, they didn't call it that just then, the tRNA chemically bound to the amino acid was needed for protein synthesis. So how did they do that then? Well, they got, so they knew that they had this stuff because they put a 14C radioactive amino acid in there. So if they got tRNA and the 14C amino acid, and they added that to the microsomes. So remember that the microsomes were the ribosomes, basically, with a bit of membrane in there. You get protein synthesis. And interestingly, they showed that we actually need GTP for that. Guanosine triphosphate. And nowadays we know, it's quite well known that in order for protein synthesis on the ribosome to occur, for the amino acids to join together, you do need GTP as the high energy molecule there. So they discovered that. They also showed that if you got just the tRNA, so if you got tRNA uncombined with no radioactive amino acid attached to it, you haven't got that on it, and you just add the amino acids separately and the microsomes, no protein synthesis. So you need this, they use the word a charged amino acid. What they mean by that is they mean this thing here. They mean tRNA plus with the amino acid attached to it. You need that in order for protein synthesis. It's no good having the RNA separately and the amino acid separately. That won't do. They've got to be joined together. And of course, to make them join together, you need to use these enzymes in that previous step there. Yeah. Okay. So by that stage, they'd shown that you do need a transfer RNA molecule joined onto an amino acid in order for protein synthesis to occur. And then the next thing they had to do was they had to characterize the tRNA molecule. Right, so they found out that this it's smaller than the RNA that they knew existed in the microsomal fraction, which we now call them ribosomes. So it's different to ribosomal RNA. They found out its molecular weight. And from that, they could deduce roughly the number of bases in that molecule. And then they started looking at the way in which the amino acid is attached to the RNA, okay? And they found out that it was attached to the 3' OH on the ribose of the last base in the molecule. Here we're saying the first base is going from the 5' going to the 3'. So they had roughly characterized the size and everything of the transfer RNA. They knew that you needed an enzyme called the amino acid acyl transferases. An enzyme was needed to get RNA and the amino acid to turn it into a tRNA amino acid, and also that you're going to need ATP for that to happen. It was left to other groups then, to get these tRNA molecules, and they digested them into smaller fragments, sequenced the fragments, put all the fragments together. And by 1968, the sequence of, I think the first one they did was an alanine tRNA that was sequenced. They realized where the codon was, the anticodon, sorry, realized where all the loops which base paired in the whole structure was sorted out by 1968. But so what we take for granted now is, we know what a tRNA molecule looks like. They're very much working in the dark, and they had to be fairly ingenious in order to work out that molecule actually existed.